Hi friends, today in my video I talk about my research. Actually in my research all we will know about how the seed breaking the dormancy, how many days, how to check the viability of the seed, how many days it takes for germination. So I selected the orange seed. Everyone will think why I selected the orange seed. Just simple friends. This is a summer season, so we want to take orange. It's good for health. At the time of eating an orange, I'm thinking how the seeds are getting breaking the dormancy and how to check the viability and how many days it takes for germination. So the simple I'm taking selecting the orange seed. This is the reason and removing the seeds from the orange and cleaning properly. And next process is all we know about seed treatment because we want to prevent from a seed-borne disease. So seed treatment I selected NaCl salt. NaCl added for 1.5 hours I am giving for seed treatment. In that 1.5 hours after 20 minutes I observed that seeds are respiration is done in this process of seed treatment. You are seeing in the picture seeds are getting bubbles, that's a respiration, so the seeds are viable for germination. Okay, after I am removing the water and keeping the fresh water, and with 24 hours I'm keeping the fresh water. After the 24 hours I'm removing the hot cells of the seed. Why I am removing the hot cell of the seeds? Because it takes germination, it takes very fast, so I'm removing the hot cells, but only one seed. I'm not removing the hot cell. I want to compare this both, how which one is getting fast germination. So one seed I am not removing the hot cell. So and selected the petri dish and I'm cutting the tissue paper for the moisture absorber. You don't want to give more water for the seed because the seed will be want to suffocation. Suffocation means the it require more oxygen. If you are giving more water, it will be difficult for respiration. So it's I'm choosing the tissue paper for moisture absorber. It always present the particular water and moisture because in the orange seed it's require the temperature of medium temperature, not much more cool, not much more hot. So it will be a very cool temperature, optimum level. So daily 3 times I'm spraying the water in the petri dish. In this tissue paper will always absorb the moisture. And for seed viability, we always check in laboratory tetrozolium test. But I done only a salt because home remedy. I'm also always I'm already mentioning to all salt. I'm checking the viability because all you are seeing the bubbles. So seed is respiring. So seed will be definitely germinate. So daily 3 times I'm spraying the water. After the final result, after the final result of 2 weeks, all the seeds get spotted. But only one seed is still not spotted, still not germinated. Why? I'm not removing the hard cell. So it takes much more process. So it takes much more time. So I observe that. And I want to mention the orange seeds are epigeneal. Above the ground. It will be grow above the ground. Okay, that's my research. I'm observing. Okay, points to remember what I done. Points to remember what I done. First, I'm removing the seeds and seed treatment. I'm taking NaCl, not tetrazoleum, NaCl, and I'm observing the respiration for viability, seed viability. No, not require any tetrazoleum test, only a salt. The seeds are getting respire. You are seen in picture. After what I done, after next 24 hours, I'm removing the hard cells and only one seed I'm not removing the hard cell. For comparing this, how many days it takes. But removing the hard cell, it takes very fast for germination. 2 weeks the process is going, my research. Finally, I observed that after removing the hard cell, it takes spotted for 2 weeks. And for the checking the viability, same seed treatment I had done the process of NaCl. Okay, that's my research, friends. I'm sharing to all. If you like my video, subscribe my video. Thanks for watching. Jai Hind.